Again, welcome back to MLT 120. Today we're going to learn how to do reticulocyte counts. Remember, our reticulocytes on a right stain appear kind of this bluish purple color. And on a supravital stain, you can see the ribosomal RNA inside of our retics. So let's review real quick exactly what retics are. Retics are immature red blood cells with remnant RNA in organelles such as the mitochondria and ribosomes in the cytoplasm. Remember, our mature red blood cells or erythrocytes no longer have a nucleus or a mitochondria. Normal reticulocytes spend about 2 days in bone marrow and about 1 day in our peripheral blood before fully becoming mature red blood cells. To see the reticulum or the inside parts of the retics that remain, you must use a supravital stain to visualize them. They will not show up on a right stain. Supravital means that the stains show the living parts of the cell. As the red blood cell matures, those parts that remain in the reticulocytes will gradually be lost and can no longer be seen in the fully mature red blood cell or erythrocyte. So here's a question. How will retics or reticulocytes appear on a right stain? They will appear as polychromatic red blood cells, meaning they're going to have kind of that bluish purple color, but you won't see any remnants of RNA inside of the red blood cell itself. You're going to have a supravital stain in order to see anything inside the red blood cell or the reticulocyte. So what's the purpose of even doing a reticulocyte count? Why would we be interested as far as a lab result? It's to determine the erythropoietic activity of a patient's bone marrow. The reticulocyte count measures the number of young or immature red blood cells in your blood. What we would expect to see in a patient with aplastic anemia is that aplastic anemia is where your bone marrow is failing, or a complete failure, pancytopenia, of all cell lines. So you have a decrease in all cell lines. So the test shows whether the bone marrow is making red blood cells at the correct rate or not. So people that have aplastic anemia would have low reticulocyte counts. So if they have low immature red blood cells, guess what? They're going to have a low number of mature red blood cells. So here is our next question. What is the purpose of performing our retic count? The answer is it's going to help determine our erythropoietic activity of our bone marrow, and it's ultimately going to help us determine whether a patient is anemic or not. Remember, our kidney is the organ that produces our hormone erythropoietin, and our erythropoietin kicks off the whole process of our bone marrow to produce red blood cells. And we must have immature red blood cells, our reticulocytes, before we have mature red blood cells in our peripheral blood. And if we don't have immature red blood cells to develop into mature red blood cells, guess what? Our patients are going to be anemic. We've talked about supravital stains and it being necessary to have a supravital stain in order to see the inside contents of reticulocytes, but there's a couple of different supravital stain options out there that you can use. We've talked about the new methylene blue in the past, and again you can see the remnants. Here's a little bit zoomed in or magnified image, but here are reticulocytes in amongst some regular red blood cells. Here's some regular red blood cells, and here are the reticulocytes. You know they're reticulocytes because you can see the remnants inside, plus they're a little bit larger. But this is the new methylene blue, and up here at the top is another type of supravital stain that's called brilliant crystal blue. And the retics appear very much the same as on the new methylene blue. You can see the remnants inside. The retics are a little bit larger than the regular mature red blood cells, but these are just a couple examples of supravital stains. Okay, let's talk about the specimen that we need to perform a reticulocyte count. We're going to use our EDTA tubes, or our purple top tubes again. We want to make sure they're well mixed, and we must do our reticulocyte counts within 72 hours of collection. This is really important for the reticulocyte counts. It's really important to not do a retic count on a specimen older than 72 hours because retics can continue to mature inside our specimen tubes. So old tubes, those retics can continue to mature, and they can fully mature into erythrocytes, or red blood cells. So when we do a retic count on an older specimen over 72 hours, guess what? Those retics that we did have when we drew the specimen are now fully mature red blood cells. So we'll get a false decreased count of retics because they're no longer retics. They've matured into erythrocytes. So it's really important to make sure you do a retic count early on with your specimen, because they can continue to mature in your specimen tube. So now let's discuss the procedure for a manual reticulocyte count. First thing you're going to do, you're going to mix equal parts of blood and stain. And the stain that you're going to use is going to be one of your supravital stains. So let's, for example, use our new methylene blue. So you're going to take a few drops of your new methylene blue and mix it with a few drops of your blood. Okay, once it's mixed, you're going to let it incubate at room temperature for about 10 or 15 minutes. Then once it's incubated, you're going to remix it together. And just make sure it's well mixed and suspended together. Then you're going to take your mixture and you're going to make 2 to 3 slides. And you're going to prepare your slides in the same method you would make any other blood smear slide. You want a feathered edge where you can read the slide, just like the image over here. You want that nice feathered edge. So now you're going to take the slides that you prepared for the retic count, and you're going to put them on the microscope, and you're going to count up to 1,000 red blood cells under the 100x objective with oil. Okay, so once you get to 1,000 red blood cells, you can stop counting. So you need to make sure you note that you've counted up to 1,000 red blood cells, and along the way, while you're counting 1,000 red blood cells, you need to note the number of retics that you see in those fields of view. Okay, so once you get to 1,000 red blood cells and you know your number of retics, you're going to use this formula down here to determine the percentage of retics. So you're going to take the number of retics you counted, multiply it by 100, and divide it by 1,000 red blood cells, because that's the number of red blood cells that you counted, to get your percent retics for that patient. For the procedure for retics, you need to count at least 2 slides. Okay, so that's why we made so many slides. You have to count at least 2. If the results of your 2 slides do not agree within 20%, the 3rd slide must be counted. So that's why we made 2 to 3 slides. The appearance of the retics on a supravital stain, they will stain blue, and the reticulum should be visible. Okay. On a right stain, they will appear large and polychromatic. They'll just be that bluish, purplish color. You won't see anything inside. Okay. You won't have the reticulum visible on a right stain. So make sure you know the difference of how your retics will look on a supravital stain versus a right stain. For our normal ranges, for the retic counts. For newborns, it will be 2.5 to 6% retics seen. That's our normal range. For adults, you'll only see 0.5 to 2% retics seen in a normal patient. So let's look down here at this image and try to determine, is this, looking at these retics, and this one down here, the ones that the arrows are pointing to, do you think this is a supravital stain or do you think this is a right stain? I'll give you just a second to think. This is a right stain. We know this is a right stain because we do not see anything inside these retics. Okay, you don't see any kind of reticulum, any remnants inside of the retics. So this is a right stain. Some things to consider are sources of error with the manual method for retic counts. Very anemic or polycythemic patients you must adjust the portion of dye. You may have to add a little bit more in order to be able to see all the retics. Blood must be mixed and stained well to see all the retics. Must remix during the staining process, or you may not be able to see all the retics. Other red blood cell inclusions will be visualized on a supravital stain, will make it more difficult to do a count correctly, so maybe misidentification. These include our Hal-Jolly bodies, our Hans bodies, our Pappenheimer bodies, and even basophilic stippling. So just be careful that you're looking at true retics and not some of the other red blood cells with inclusions. We do have a device called a Miller disc that we can place on the ocular of the microscope to help facilitate counting our retics, because it is kind of difficult to make a slide and manually count 1,000 red blood cells. So we do have a device that allows technologists to count less red blood cells. With the Miller disc, the retics in the larger square are counted and the red blood cells in only the smaller square are counted. And we're going to see what the Miller disc looks like, or what you'll see through the Miller disc on the next slide. So this is what you would be seeing if you placed a Miller disc on the ocular of the microscope. You would make the slide as normal, but you would have this box A and box B, or the larger box and the smaller box, and you would count all the red blood cells in the smaller box, or box B, and you would count all the retics in the larger box, or box A. And then you can use this formula that goes along with the Miller disc in order to come up with your percent reticulocytes for the patient. So it makes it a whole lot easier than counting up to 1,000 red blood cells, because you're only having to count the number of cells in those little boxes. Okay, so we've talked about the manual methods for reticulocyte counts, but most of the time our reticulocyte counts are done through automated methods on our hematology analyzers. We use lasers on the hematology analyzers in a process called flow cytometry, and we use supravital stains to perform the automated retic count. And through the process of flow cytometry, we can differentiate between older versus younger retics, and we can also report both the percent retics and what's known as the IRF, or immature reticulocyte fraction. It is useful with patients when they're on treatment to increase bone marrow activity, so we know if the bone marrow is actually producing our red blood cells, so we can know if you got immature red blood cells, then hopefully eventually you're going to have mature red blood cells. So let's look over here at our image of how flow cytometry works in our automated hematology analyzers. So here is our sample of our blood going into the instrument. So we have all types of blood cells going in. So we have red blood cells, we have white blood cells, we have retics. All those are mixed in together going into our tube. So we have this nozzle, and it kind of gets narrowed down until it's only one cell thick going through a tube. And we only want it one cell thick going through because we want it to pass by this laser only being one cell thick, because we want the laser to hit each individual cell. Okay, once that laser hits each individual cell, depending on how that light is scattered on that individual cell, it will tell the instrument, hey, this is a retic. Hey, this is a mature red blood cell. Hey, this is a white blood cell that's a neutrophil, or it's a eosinophil, or whatever. But in this case, we're looking for reticulocytes. So when the light is pointed on the cell, and depending on how it's scattered, it should identify it as a retic. So that's how we're going to get our reticulocyte count, using flow cytometry. Each individual cell will have a laser pointed at it, and depending on how that light scatters off of that cell, it's going to be identified. This concludes our lecture on reticulocyte counts. I hope you learned a lot about the manual methods and also the automated methods. If you have any more questions, don't hesitate to reach out through email or during lab time.